We don't have a crystal ball, so we need to push everything that could possibly move the whole field forward. Here's my motivation. This is Andy. He's 36 now, lives at home with Mike and me, and we started FRAXA when we first learned about Fragile X, which he has, and just thought, it was scary to think of his life going forward. So here we are, 33 years later, still working on it. FRAXA has funded more than 35 million in research grants in 19 countries now, and 675 or more annual grants so far, because that's what it takes to make progress. And here we are, close to the first big, exciting potential fork in the road or breakthrough, we hope, this fall, because there are two pivotal clinical trials that are scheduled to finish in the coming months, and we expect results from both of them in the fall. Zatolmilast by Shionogi, used to be Tetra, is on target. The adult trial will wrap up first in the next couple of months, followed closely by the adolescent trial. And Harmony's study of Zygel, ZYN002, is pretty much on the same timeline. Really, if either one of these succeeds and goes to the FDA for approval, then we not only have an approved treatment for Fragile X, the first one ever, but also a path forward for other solutions, because we'll have clinical trial designs that we know work, for example. So it's exciting. We have to wait because the data is blinded until the last patient finishes, and all we can do is cross our fingers. But in the meantime, based on what parents want, FRAXA has established a Curative Therapies Fund where we are targeting the root cause of Fragile X. And just a poet's refresher, if you will, on the root cause of Fragile X. You have a gene defect in the FMR1 gene. That means that it cannot produce its protein product, FMRP. Normally, genes are transcribed into mRNA, messenger RNA, which can travel from the nucleus of the cell to other parts of the cell where they affect translation of a protein. So in Fragile X, no protein. And that is the root of the problems. So one of those many grants that FRAXA funded went to Dr. Joel Richter at the University of Massachusetts Medical School, and his team, his collaborator, Elizabeth Berry Kravis at Rush University Medical Center. And we've been funding this team, and both of them separately as well, for several years. They made a serendipitous discovery that the Fragile X gene is not entirely silent as was thought. In fact, it produces, in most people, it produces an abnormal version of the RNA. And that abnormal version still is unable to produce protein. Let me pop back to this slide for a minute. So we always thought the problem was here, no RNA and no protein. What Dr. Richter found is that actually there is RNA, but what he found in most patients is that it's a splice variant, a defective version of RNA that cannot produce protein. So he went to his colleague, Dr. Jonathan Watts at UMass, who is a master ASO expert. And I will say what an ASO is next. But anyway, Dr. Watts created ASOs. The team put that into the dish with the cells. Lo and behold, it blocked the RNA that was abnormal, and the cell responded by producing normal RNA and normal protein. Here's a little graphic of it. So here's your gene in the nucleus, which apparently in the case of Fragile X is transcribed into a message RNA, which goes out of the nucleus into the rest of the cell. If you introduce this ASO, which is a short engineered single strand molecule that matches the splice variant, the defective RNA, like a puzzle piece would match. It latches on, binds it up, basically neutralizes it. And then lo and behold, they found normal RNA produced and normal protein. So this was so exciting that we held a Banbury meeting, a research meeting, a couple summers ago, and Dr. Richter presented this to everybody, and plans were made, and now we are at a point where pharma partners have signed up to take this forward with the goal of getting to the clinic. Why do we need pharma partners? The cost goes up extraordinarily. So FRAXA can fund the research, the academic research, but you absolutely need a pharma partner that can invest millions to do the next steps. And so Curative, a Cambridge, Massachusetts-based pharma company, licensed the discovery of the Richter lab, and they are now working to develop just the right ASO for Fragile X. And they're working with a second company, also starts with Q, Quiver Bioscience, which has some super cool optical genetics technology to view how cells respond when you put an ASO into a cell in a dish. They have this technology where they tag the cells and the cells flash light when they're active. We, FRAXA, are funding part of this work now. This company, Quiver, is a startup, and they don't have the funds to do all this work themselves. So this is a new kind of grant for FRAXA because it's going to a pharma company, not just a university. So we're really excited about this work. We are also continuing to fund the Richter Lab because they are doing more of the work that's necessary to bring this to the clinic, specifically working on biomarkers. So you can tell if a treatment is working in a live animal and ultimately a person. You need biomarkers to do that. Another curative therapy that I want to highlight today is the work of Dr. Jeannie Lee at Harvard Medical School. Now, we've been funding the Lee Lab for 5 or 6 years now. She is a real leader in the field, and she discovered two different ways to reactivate the Fragile X gene in cells and in a dish, that is not. And one of those ways uses the CRISPR system that's been modified so that instead of actually editing the Fragile X gene, it goes in there and triggers a cell's natural DNA repair mechanisms. So it kind of tricks the cell into chopping some of the extra CGG repeats that impair the Fragile X gene itself, rather than doing something as scary as editing the gene directly. And the results that she found with our earlier grants were that this works and that the cells then produce normal protein. So this approach is very exciting. It's a little bit earlier in the process than the Richter lab approach, but the goal is the same, to do enough of the work to get a pharma partner to invest the millions needed. So Dr. Lee has managed to win, just at the end of last year, $1 million award inside of Harvard Medical School to take these results and test them in Fragile X mice, which have been engineered with human brain cells. So I won't go into the details of that, which you can find on our website. But investors want to see this work in mice, in live mice. They want to see that the gene can be reactivated and basically reverse symptoms in those mice. And because she has that award, that leaves FRAXA open so that we can now fund the next step in this research, which will be to take it into organoids. So there's a progression in research. You try something in cells in the lab, you try something then in live mice, and more recently, we've been really lucky to have a new model emerging, which is organoids. Now, this is a little bit of you've probably heard of organoids, but if not, they are three-dimensional balls of cells, very small, like a pinhead, started from a human cell, actually a human blood cell, which has been then programmed back into a stem cell state and then grown kind of forward into neurons, which connect and communicate with each other. And they actually form mini brains with different kinds of brain cells and different kinds of networks. So it's really an incredible platform to be able to test treatments. This is also a way to personalize medicine. So organoid could come from my son Andy's blood or your son's blood, and then grown up into a little pinhead, and then cured or treated with various approaches. They can also create a twin organoid that doesn't have the Fragile X mutation. So you have this pair of organoids for a single individual person. So FRAXA has funded quite a few studies using organoids over the last several years, and more now. And this is the model that Dr. Lee is going to use for this FRAXA grant that just started. The goal here is to take this forward, as I said, so that we can get it in the hands of a pharma company and take it forward, hopefully, to the clinic. Having just said that we don't need to rely on mice, I actually want to highlight another project we're funding, which is based on mice. So we have lots and lots. Think 100 different potential treatments that need to be validated. They started, perhaps being validated in the cells in the dish, and eventually we want to get to humans, but not until we're sure that this is the right treatment target. And so mice have always played a role in this, but typical mouse studies were, in single mice in a cage, and that does not model mouse behavior. Mice live in groups, they have complicated social activity. And so this team in Belgium with FRAXA funding for the last 3 or 4 years has developed a live mouse tracker where the mice wear these chips, kind of like the chip for your pet, so they won't get lost. It's a radio frequency chip. They wear chips and then they go about their business in the cage in groups. And there are sensors all around the cage that just track what they do without disturbing the mice. And they can test treatment approaches in this setup. So the live mouse tracker is one of the resources that we provide. There are others. We have a mouse testing facility in Chile that's been funded for over 10 years now that has tested almost every treatment that, is being considered for Fragile X. And this will join in the mix as a more sophisticated platform. So between the mice, the organoids, and some other technologies like CRISPR, science is moving much faster than it was. I encourage you to go to our website, to the research section of our website, and select the type of research you might be interested in, such as curative therapies, or the university you might be interested in. There are hundreds of them. And browse around, see what's been funded, see, how it's moving forward. We don't have a crystal ball, so we need to push everything that could possibly move the whole field forward. And it really has. We're at a point now where hopefully we have a first treatment approaching. There are many others being developed, being considered. We're working with a lot of pharma companies right now that are really waiting to see what happens with these trials. And there are better drugs on the way, meaning better, meaning more potent, less side effects. And so I'm really looking forward to the coming year.